So I'm just going to very briefly introduce the next session, which is the clinical panel. Can I get all the panelists to come up slowly and take a seat at the front? But like we were saying yesterday, this is going to be a discussion, a clinical panel discussion, which is really something we want a lot of your input into. I'm not going to introduce the panelists themselves. I'm going to get them to introduce themselves. I need to do a whistle like Paul, but I can't whistle. Thank you. Very effective. So we're going to have a clinical panel discussion. So like we were saying yesterday, this is a really dynamic discussion. We're going to talk about all the things you've probably just heard about, the inequities. We're going to probe some of those things with people that are really involved in this area of expertise and of lots of different voices. So I'm going to ask the panelists if they don't mind to just introduce themselves briefly, just to kind of make sure that we all know who you are, although we all know who you are. And I'd just like to quickly introduce our moderator. So Isk, is he up there? Could be Leonard if you want. But Isk is really kindly joined us. He's an ABC. He won't tell me a lot about himself, but he just said to say he's an ABC journalist. He's on the news and the radio in Victoria, and he is, I think, a really good interface between M&D community and clinicians and researchers, and so he'll really try and bring this discussion together. So we're very thankful to have you. I'm going to turn it over to you.
Thank you. Yes, I read the news. There's no teleprompter here. Please bear with me. But I think it's most important that the panelists all just say who they are, just to reintroduce themselves. The microphone should be on. Let's start at this end, please.
I'm Jeff Rothstein. I'm a neurologist and neuroscientist at Johns Hopkins. I've been following ALS patients and running clinical trials since 1990 and run the ALS clinic there.
Matthew Kiernan, Director of Neuroscience Research Australia and neurologist based in Sydney doing clinical trials.
Hello, I'm Pam Shaw. I'm Professor of Neurology at the University of Sheffield and Director of CITRAN, which is the Sheffield Institute for Translational Neuroscience. So like Jeff and Matthew, I look after ALS patients, and I've done lots of clinical trials for several decades as well.
I'm feeling a little overshadowed. I'm Kate Johnson. I'm the CEO of MND Victoria. I'm also on the board of the MND Collective and the board of Palliative Care Victoria.
I'm David Berlowitz. I'm a respiratory and sleep physiotherapist, and I've been doing, I'm also a clinical trialist, particularly in non-invasive ventilation and models of care and other things in motor neurone disease for about 30 years, and I'm a clinician.
I'm Natalie Gauld. I'm a pharmacist with a PhD, and I've done a reasonable amount of research in areas such as gout, Hep C, HIV, and I'm on a very steep learning curve with MND.
And you've just met me. I'm a neurologist here in Melbourne and involved in clinical trials and the MND Research Collective.
Thank you to all of you to spend this time. We are hoping that we have lots of questions about clinical trials and care from everyone. There are no dumb questions. It's an amazing panel. But I did hear something interesting about amazing panels and intelligent audiences. When you say that, most people feel too scared to ask a dumb question. So that's maybe why I'm here, because I'm Iskandar Razak with a Bachelor of Arts degree. Honours in philosophy. Impressive? Yes. Well, with that background, my first question, realistically, from a layperson's term, there's no cure, but how long until a clinical trial has a cure? Raise your hand if you think it could be in the next year. I thought we'd have an exclusive story tonight. Okay. How about in the next 2 years, 5 years, 10 years? 20 years, 50 years, 75 years. Yeah. Realists, 100 years. And cure means by any definition of the word that you have in your head. Yeah. And well, okay, let's define cure by anything that either stems, stops the disease and or reverses it, or finds the cause and can be preventative.
Stems exists already. To a person, and any one of us could have probably answered this, or some of us, but gene therapy for a subform of ALS, and I think many of the audience know this already, clearly has profound clinical effects, slows the disease, and at least what I've heard indirectly from my colleagues, 50% of patients show some improvement. That's unheard of in a disease like ALS. So that's exciting for all of us that run trials. Will that be replicated in sporadic ALS, which is the core to some of Virk's questions? And I won't speak for the others. I rarely use the word cure because I don't know enough scientifically on how to do that. On the other hand, 20 years ago, 30 years ago, I don't think we would have ever expected SOD1 biology to come out and dramatically slow the disease. There's no question it's replicated. In the next couple of years, I think FUS gene therapy will do the same. But beyond that, I think you have to be really careful extrapolating science that hasn't been discovered yet.
So there could be a series of treatments that are effective. Dame Pamela Shaw, I've actually seen you speak about this on the BBC as part of my research.
Yes, I think we're all wary of the word cure for such a complicated disease. My view is that I think tofersen is a real trailblazer. It's shown us that if you get the right therapy to the right subgroup of MND, you can have really dramatic therapeutic effects. That's been a real trailblazer. So has the fact that neurofilament levels may be at last giving us a biomarker that the treatment we're giving is working, is preventing motor neuron death. We haven't had biomarkers of therapeutic efficacy until now. The fact that we may have one, and it needs further work to confirm it, means that we may be able to do clinical trials, test therapies more quickly, but also when we put a group of ALS patients into a trial, and the overall result doesn't look definitely promising, we may be able to pick out responder subgroups. So I personally think it's a time of real hope. My view is that we'll probably make step-by-step progress in slowing disease progression, catching it earlier, directing the right treatment to the right subgroup of patients. But we're not there at I don't think we can project a time scale for an absolute cure.
Does anyone on the panel want to add anything to that?
Please try really hard.
Yes. Well, clinical trials, what can be done when it comes to improving clinical trials then? Anyone want to add?
Well, I think Pam sort of touched on this, that we have typically just taken a whole group of patients and started them on a therapy without really understanding the patient groups very finely. And I think that the more that we can enrich a group, but also homogenize the patients. So try and choose patients who are more likely to benefit from a particular type of approach and monitor them with a biomarker. Then that has started already now in general clinical trials. That's going to increase the likelihood of success. I think also the meeting that Hiroshi Mitsumoto organized in Airlie House, which indicated and recommended that all clinical trials should incorporate a biomarker aspect, so that even if the trial is a negative study, we get more understanding about the mechanism of the disease or some part of the disease that's going to inform a future trial. So we're sort of future loading the approaches and gaining regardless of the results of the trial. Obviously, all of us want a positive outcome for a clinical trial. And that's how we go into it.
So will there soon be just clinical trials based on biomarkers?
Well, I think that that's the way that the field is moving. I mean, traditionally, the initial trials were based on survival, and they're very long trials. And I think in our heart of hearts, of course, we'd like to have long trials to try and work out every single aspect of it, but we have to be practical, and patients don't want long trials. So we need to try and get the trials as short as we can. And the only real mechanism of doing that is through a biomarker approach.
However, I'm going to go against what you just said. I do not believe today that a biomarker alone should define a positive trial. At the end of the day, it's how the patient's doing. Now, at some point, if the biomarker clearly and reliably predicts the clinical outcome, then yes, but that does not exist yet. I was excited as we are about neurofilament. It's not that strong in terms of predicting. Someday, maybe it will be. So you can't, and there are companies that think the biomarker is everything. They don't know what they're talking about. People who've seen ALS long enough understand that better. And still, at the end of the day, it's what the result is clinically. And biomarker may help us, may fine tune, may make trials a little easier to start with. I mean, to be fair, I think Pam and I are maybe, I don't know if Ludo or Angela, there are only a few people who actually ran the original Berlowitz trial. And by today's standards, if we had run that trial today, the way we run trials now, we probably would have never found that drug. They're short trials, and maybe we got lucky. What's happened over the years is, and I'm not really a trialist, is the sophistication of trials has changed since 1995, 93, when Berlowitz was run, to now it's much better. I would argue the science has advanced far more than trial design, but and that continues to improve. And there's controversies how the Europeans do it, how the US does it. I won't get into that controversy. The man over there likes to poke. And that's fine. That's no different than lab science is controversial. So can be trials. At the end of the day, we want the best trial design to get us the answer ideally as fast as possible, but we need a good answer. And we don't want bad trial designs to miss a good drug. So.
Two things. Has anyone ever said that you look like Michael or Kirk Douglas? Secondly, I think David has something actually important to say.
I really doubt it's pertinent, and it's not going to be that funny. I also want to put in a plug for more creative trials. I think there's, in many ways, we're kind of hostage to fairly old-fashioned designs and lots of things, notwithstanding things like the Healy platform trials. But there, people like Pecori in the US have developed really, really innovative emulated RCTs within cohorts where one can control for exposure biases in non-traditional confounders, such as, in my case, NIV kind of dose exposure, not just a binary variable, but if it really affects survival. And there, in all the cohorts, there's a dose response. It's probably important to deal with, but we don't deal with it in the S-demands for most of our traditional trials. So I think some work needs to be done around embedding traditional pharma therapeutic kind of trials within a broader cohort kind of design that can take into account different environmental and other kind of exposures that not only interact, but interact differently over time. So I think there's work to be done that many of us have ducked around being a bit more sophisticated about how we mush our trials together with the rest of the real world.
Just having been part of the MND collective and working with Julie on the Lived Experience Network, there is a lot of talk about the need for better communication about which trials are happening, giving that information to people living with MND and their families, and also communicating along the way what the outcomes have been so that they're aware that the trials are happening and what the impact of them has been.
Just checking, any questions from the audience about this? Let's try and find. I can point and you can shout. I can try. I'll run out.
So I think most of us think that one drug will never substantially make a difference and that we need combination therapy. Yet the pharma model is very much looking for that blockbuster drug that's going to make them a lot of money. Monotherapy trials. I mean, that to me is a flawed approach. How do we change that?
I would argue, and we'll argue this in the debate shortly, that before we do need to tackle multiple mechanisms, but before we start putting drugs together, I think there has to be a signal that that single drug is doing some good. And then we put them together when we've got several of those.
Yeah.
We're already doing that because most trials, patients are on Riluzole at baseline.
So for an audience, I mean, some people in front know this, for the audience, almost every trial we run today is already a combination. It's Riluzole plus another drug. We never run, it's unethical, at least in the United States, to run a new drug without giving an FDA-approved drug. So they're already that combination. To be fair, though, I always have this discussion with patients because they always think, can I add, can I add, and I add? And there's two very good examples I know of. One, years ago, Novartis ran a trial, I can't remember the drug, with Riluzole, and they were excited about this new drug, as most companies are, and we are as well. The drug didn't work. The drug plus Riluzole was actually worse. So putting drugs together without knowing some of the biology, and that was not predicted by any preclinical. And of course, the second, again, for an audience that doesn't know, is even drugs on the market, because there's a lot of push for taking existing drugs. Couldn't that be great? Couldn't they be safe to take? They've already been given to millions of people. Minocycline, which was an antibiotic used extensively by hundreds of thousands, not millions of people, in a model, in the SOD1 model, was quite effective. 3 independent labs said it was, at the time, this is 2004, so best drug ever. In ALS, this was a single site trial at a Columbia University, it sped up the disease quite significantly. So just because a drug exists rate does not mean it's always a good drug to use. We have to do due diligence, and adding drugs together is exciting, but there's got to be a lot of science that defines it. And maybe biomarkers will give us that information that we never had before. I don't know, but it's not a simple add two together. She's going to talk more about it.
I suppose also the polydrug idea suggests that there's multiple processes involved, and that's a theory, a hypothesis, but we don't really know. We don't know strictly the cause of the disease. And back to Amar's talk yesterday, we don't know the match that's lit the fire. And surely, I mean, a lot of pathways are inevitably involved, but are they causative pathways or are they adaptive pathways? And that makes it quite tricky until we know the exact process of motor neurone disease. So mechanistic insight, we're always going to be shooting in the dark.
I do want to make sure that people away from this. Let's go to up the back here. I knew I wore these boots for a reason today.
I guess the main takeaway from the last couple of days is kind of the heterogeneity of the disease. And we've had comments about how an effective therapy will probably be a combination of a couple of drugs. You said before that before we can find any effective combination therapy, we need a signal that it's doing something good for it. How confident are you that there is one signal that can measure that good across all the therapies that you try? Does that make sense? Have I made that correct? Do you know what I mean?
So I guess the signal that you're looking for, I think we need to establish target engagement, that the drug you're trying is getting in sufficient concentration to the right place in the nervous system. So that's one important point. And the signal you're looking for might depend. And I think we've learned a lot from tofersen in relation to that. So you could see evidence of target engagement, which was a reduction of the SOD1 protein in CSF within 8 weeks. You could see the neurofilament levels coming down within 12 weeks, within 3 months. But at 6 months, the clinical benefits weren't evident. We had to wait 12 months to be able to see that. So it depends, you know, there are different types of signals. Some of them might happen quickly on the basis of biomarkers, but to detect clinical evidence of benefit, you have to wait longer. But there may be readouts that can give you a signal earlier than waiting to see, as Jeff said, that the patient feels better or feels that their disease has slowed down. So I think it depends on the agent.
And another, this is why it's so complicated. So the timing, so maybe some of you love inflammation, others may love oxidative stress. Well, we don't know when that happens in the patient. And maybe a biomarker may pick that up, but even if it does, you love your pathway, but maybe in reality, it contributes to 5% of the true injury in a patient. You could have the best drug and it only contributes to 5%, meaning high efficacy drug, barely touch the disease. So we bring our science bias to the pathways, but we don't know really in a patient how much of that disease at the time we see a patient is being driven by TP43 misfunction, oxidative stress, inflammation. And when we see that patient, which of those are more active? So all of those variables over time, when you see a patient, make it extremely complex. What will happen for the field will be, do we at least have biomarkers, some way of measuring each of those pathways? So you can fine tune, but it still may not be. You may have a great biomarker for inflammation, you know, IL, whatever, yet it doesn't really matter because it's not that potent. And that's something we don't know much about in patients. How potent is the pathway? We know a pretty weak drug like riluzole, has a small effect, but we've yet to see other effects in other pathways. I want to add one other fact. I hate doing this, but it's because a lot of this audience is not in the clinical. Many of you get excited. You read news reports by biotech companies about positive phase 2 trials. Oh, isn't this really exciting? A real fact, at least 80% of positive phase 2 trials, at least 80% in neurological disease fail in phase 3. So just statistically, when you see a positive phase 2 trial, it's likely to fail. It's just, that's medical fact. And that's not unique to neurology. It's seen in other fields as well.
Natalie, actually, Sue.
I was just going to ask the room whether you think there's enough collaboration between, neuroscientists, clinicians, and big pharma, as well as patient groups, in the lead up to developing these big and expensive trials, or are they going ahead with their own internal ideas on how this should work and wasting time and money and effort?
There's increasing work in that direction. We heard from LifeArc before lunch, and they're very much bringing those elements together. In the UK, we've formed a national M&D research institute that has patients at the heart of it, advising about what's going on, bringing together clinicians, clinician scientists and basic scientists, working in different work packages. So I think the voice of patients, my perception anyway, is that we're taking notice and taking advice. We've had a patient and public advisory group in Sheffield for 10 years, and every study we do, they give us their opinion, they put forward their priorities, they modify documents to make them better understandable for people living with MND and their families. So it's not perfect yet by any means, but I think there's an increasing thrust towards getting those people together. My perception in terms of pharma and biotech companies is that often they don't run the basic science themselves so much in the way that they used to. So they're very interested in collaborating with academic institutions that are doing the basic biology. That's my experience in the UK.
In the
And
United
also-
States, very sort of similar. We have a group called NEALS, Northeast ALS. It's not really the Northeast, and Dr. Bowser and I are both on the advisory committee. Almost every company, not all, presents before they do their trials, their preclinical science to this committee, their clinical trial designs. We're advisory. They often don't know, actually don't often have a real depth of the science of ALS, and certainly not the clinical trials. We advise them. It's still their drug. They own it and they can do what they want, but we try to get them to do what we think is the appropriate approach. And one of the biggest discussions always, and Pam has mentioned this, is what's your biomarker? Do you know if your drug's working at target? And Bob may correct me, but my God, at least 50% haven't considered that, don't have it yet. And we seriously advise them to have that data, but it's still theirs and they'll run the trial the way we want. We don't turn them away, at least not for that reason. We do turn away sometimes companies, but not for that reason.
Couple questions. I'll go about that, but while I'm going up there, okay. Natalie, just from your perspective, when you're hearing all this, I mean, you're living the situation. What are your thoughts? And of course, what's happening in New Zealand?
So I forgot to say on my introduction that I work part-time for MND New Zealand as well. From an MND New Zealand perspective and patient perspective, we actually don't care if we get asked or not. We just want the trials in New Zealand. We've had one recently, and it was we felt lucky to get it with 20 patients. I used to be principal pharmacist at a clinical trials organisation in Auckland, and we've done world-leading research there. We were always the very first site to randomise with all the Hep C work that we did every single time. And as principal pharmacist, I always found out that there was problems every single time with them not having their flags in the right way in the machine. So you try to randomise and there'd be a problem. But we were always quick with ethics, had really high quality, and it was accepted around the world. So we would love to be partnering with others in Australia and others elsewhere, and arranging research in New Zealand. And we've actually got Sarah Buchanan here today, neurologist, who would be very keen to join in some discussions as well. So thanks for the opportunity to say that.
Do you want me to go? I'm not a scientist at all. I see so much good research happening and so much money pumped into research with support from the drug companies and all of that. My question is, how do we get more money into healthcare?
Anyone have the silver bullet solution for more money into healthcare?
Okay, as somebody who used to have a healthcare job and now I just do research, I'm going to stick my neck out. I think transparency is really key here. Not about what we say we do, but what about we actually do. NDIS above and below 65. You know, it's criminal that for the same disorder, the standard of care is functionally, structurally different in this country. Yeah, there's a whole ton of these kind of barriers. And while it applies, I think, to the entire Australian population, I think MND is the pointy end because things change really fast and they're really pretty bad. So I actually think that one of the keys around healthcare is not just money into healthcare, it's visibility of delivery of healthcare and effective mechanisms to highlight the difference between what we say we do and what we actually deliver. And most of that data is administrative, like we could do it. So, yep, there you are, fixed.
I would say what's helped in the UK is providing evidence that a healthcare intervention is beneficial for patients. So, for example, non-invasive ventilation, we had to do those really difficult trials to show the benefit, but then NICE, our National Institute for Care Excellence, produces guidelines that this is what you should be doing for MND, and that's allowed a lot of centres to develop their healthcare and the excellence of it. And also the charities, I think, have a huge role. In the UK, the MND Association has supported specialist clinics, 21 of them around the country, and put some resource into helping those clinics deliver good care. So I think clinical research showing the evidence as to what high quality healthcare can do, tackling the troublesome symptoms and providing an evidence base that tackling those symptoms does some good in terms of quality of life, life expectancy sometimes. And then you can be like a barrister to the funding bodies. Look at the evidence that this works, so you've got to invest in it.
So I want to build on that because I haven't been back here in a long time. And there's a dramatic increase of high quality research happening in Australia now in ALS. And as best I can tell, that's money based. It's not that you're all not all brilliant, but it's hard to get grants. And I don't know what the funding level is, but the organizations here have really pumped a lot of money, and that shows. And you can turn to other government officials, especially when you're talking about, well, how do we, on one end, it's the care of the patient, but ultimately you start by asking about a cure. That's going to come from a drug. Some drug one day, I don't know when. And that's the research that people here do. And to show that that boosts the money, and someone here can probably map it out, and the amount of research that's coming out, high quality research, that teaches you, I think, that that investment will get effective therapies. And so it's the whole bridge. You've heard about the clinical end. I would argue just as importantly is the preclinical end, getting that research. Because again, drug companies for the most part don't find the drugs for ALS. We do as academics. We find the targets, they build the better drug. They don't usually find that original research. We do that. And that means people here have to do that. And the philanthropies do that well, but a government should be much part of it. You can turn to the government and say, look at the progress we've made. We did it on our own. It's now your turn to do it. By the way, when I'll leave it at that.
I think the other thing is that some of the significant funders are looking for sexy things to fund, creative things to fund, excessively innovative things to fund, which means that those of us that are just trying to meet people's basic needs, like those of people over 65 for support, equipment, you know, being able to get out of bed in the morning, there's a big gap there. So, you know, if I can't get out of bed or get out of my house, I don't really want an app. I want a wheelchair.
So I've got a question over there. And
Yeah.
another question over here.
I have a question for someone online, but I also want to respond to that question too, Julie, because I just want to add that I guess we have known that NIV works for quite some time and that there is good evidence, and yet we still have inequitable access to and uptake of NIV across the country. So I think we don't just need to provide the evidence that it works, we also need to do the implementation research to understand why it's not being implemented and to address those problems. But I'm now going to read my question from Keith Melpress, who is with us online from Queensland. And Keith's a legend, he lives with MND, and his question for the group, which I think David has started to answer, is research or trials currently require sufficient numbers of people and duration of participation to generate confidence in the statistical outcome. Those numbers and longevity are not readily available among the rapidly changing and small group of people with MND. Is it possible to design a new approach to this to clinical trials that's better aligned to the reality of MND, that can produce meaningful results that are outside the current paradigm of clinical trials?
I think biomarkers are one important aspect of that. If you look at a disease like multiple sclerosis, when I first started training in neurology, all we did was give steroids for acute relapses. But because there's something you can measure with imaging, so you can measure the areas of inflammation in the nervous system, there are now 12 or 13 disease-modifying drugs for multiple sclerosis. And that development has happened because there is a biomarker readout of whether the treatment is working or not. So I think we're just on the cusp of that with neurofilaments in ALS. We haven't had a biomarker of therapeutic efficacy. We need more work, and we're not ready to throw out a drug because it doesn't affect neurofilament levels. But I think we're on the brink of having those biomarkers. And as we work to subclassify MND more and stop treating it as one thing, because the upstream cause differs in different subgroups. So we need to learn from cancer doctors, oncology. We don't treat breast cancer as one thing nowadays. We divide it into its molecular subgroups and target the right therapy for each patient. So that's a long-winded answer, but I think really having biomarkers that show us whether a treatment is having some positive effect at an early stage is going to partially answer that question.
We've got a question from over here.
I think that the first question really knocked a lot of us out. That was a king hit when, you know, the question was asked, is there going to be a cure in the next little while? If I understood the answer right, you'll all be dead before there's a cure for MND. Is that right? All of our expert researchers at the day, like you've talked about my death for the last 2 days. So I thought I'd bring your death into the conversation, you know. But it does sound like that. It was a king hit. It was like, you know, I don't mind a cure not coming in my lifetime, but to think that it's not going to come in the next 25 years, 30 years, or whatever it might be. So why don't we change track and put more money into care, but also making the patient more healthy? Well, you talk about inflammatory might only add 5%, but that's 5% better than nothing. You know, gut health might add another 10%, but that's 10% better than nothing. I'm probably the healthiest person in the room, apart from MND. You know what I mean? Like, I might not be able to run farther than you, but, you know, you could be dead tomorrow because of some other ailment that you have that you don't know of. So why not take the MND patient and say, look, we don't have a pill, but let's make you the healthiest specimen that you can become while you do this journey. And the cold that you get won't last as long as your wife's cold. You know, you won't end up with pneumonia in hospital, and you'll have not long life, but you'll have a quality of life because we'll give that to you.
My view is we have to do both. So take the troublesome symptoms and do better for them to improve the quality of life of the person. And I accept that, you know, in cities versus rural areas, access to devices and things that are needed in the patient's home appears inequitable and difficult to get hold of. And I wouldn't want you to think, I'm not too gloomy, but I am cautious about raising hope too much in terms of talking about a timescale for a cure. But I do believe there will be step-by-step progress in slowing the progression of the disease. What makes MND, I think, so frightening for many patients and families is the speed of change of that disability. Some people have much slower progression, which is great, but that's how I see neuroprotective treatments going. I think we'll make step-by-step progress in slowing disease progression, but it'll take a long time before we can talk about an outright cure.
Yeah, you know, everything you said, good neurologists, they do that. They care about your nutrition, your activity. So we don't ignore that. The heart of what you're asking for, the starting question, some of us have been doing this so long. So when I was starting out, it's like, oh, cure. Oh, I think in 5 years. 5 years later, that patient's not there, but a patient again asks it. And when you've done this enough, you realize I shouldn't put a timeline on it because I'm lying. I don't know that answer. So that's why we're giving that honest answer. I don't think any of us really know. We all want it. And the most exciting thing, I'm just reemphasizing this, and you've heard from Pam and others, is that the fact that we have drugs now, very specific for a subset of people, not everyone, that have remarkable efficacy in our world, that tells us we'll be able to do it. We just don't control that timeline. We know the trials that have to be done. We know there has to be more science. We know how to do those. We have to accumulate it. And to be fair, that requires not just one or two of us, roomfuls of really intelligent, creative, aggressive people who work 24/7. You fire postdocs who don't want to work on weekends, things like that. Not kidding, actually.
But I think Phil's point is even more important if we slow the disease down, because we're going to have a lot of people living longer, but with disability. And that's a hard ask. You know, I think when you talk to your patients who have slower disease, they often get round to thinking that themselves, you know, I don't like living like this, and I'm living quite a long time like this. Yeah, no. But I think well-being of people and their families is a very important thing to consider, which is what Phil's saying.
Yeah, just to support that, we should be investing much more in care. We say until there's a cure, there's care, but we don't have the resources to provide all of the care that people need. Yet we know that multidisciplinary care is one of the most effective ways to support someone with MND. If you are diagnosed over 65 and can't get to the NDIS, the most money you get from the government through an aged care package is about $52,000. On the NDIS, the average package is $242,000. So there's a massive inequity. We need to be lobbying to make sure that everybody gets all the care that they need and has the best quality of life that they can for as long as possible.
Something that happens is that we have delayed care. The government's going to incur the same cost for a wheelchair, whether it's delivered in 3 months or in 6 months. And I think one of the things in New Zealand is that we have regional variation, just as you're having variation rural to city. And we need to have audits done to see what's going on, what is actually, where could you put something in more timely that has no extra cost, but it's just more timely, that makes a big difference to somebody's life. There's a whole heap of areas in which we can improve things for patients without it necessarily being a huge cost to the health system.
We've got a question about guidelines in the UK.
Thank you. Yeah, just wanted to make a comment about what you said, Dame Pamela, about the NICE guidelines. We don't have guidelines like that in Australia. And it kind of goes to what Julie and Kate were also just saying. Look, I think if we need to invest in that to enable us to be able to benchmark best practice, to be able to give the best possible quality of care to people. So just a big shout out to any people that have got big pockets in the audience, any pharma companies that might have some money. Please, we need this desperately.
NICE has been very helpful in terms of supportive care and making recommendations as to what clinics should be doing. But when it comes to Tofferson, they haven't been so helpful. They've been dragging their feet, whereas it's been approved by the FDA in the US and the EMA in Europe. NICE is still trundling away, trying to put it down the wrong pathway, so that at the moment they're refusing to treat SOD1 MND as a rare disease, whereas there are probably only about 100 patients with that in the UK at any one time. So they've been helpful in pushing the standards of clinical care, but less helpful in terms of new neuroprotective therapies, I would say. But yeah, and I don't know whether there are major differences in the different states in Australia. I don't know enough about it, whether the state system.
Yeah, well, look, by means of update as well, there is interest in creating national guidelines. So there's going to be MND Australia with Fight MND, and they have resourced this, and they will start discussions with key stakeholders. So that is going to happen. And Pam is right. Unfortunately, we do a state-based health system, whereas aged care is a federal health approach, and this has also created some difficulties and inequity in the system. Just to go further about the points about the standard of care, we know that that's getting better, and obviously the supportive approaches, including non-invasive ventilation that Pam and her team pioneered through clinical trials, that does put difficulties then into future clinical trials. So back to Keith's question, can we come up with a new model? It's very hard to get away with not having a placebo group, because every trial we see, the placebo group is doing better and better and better. And if we compared the trial outcome with historical controls, many of the trials we've done in the last 5 to 10 years would have been incredibly positive. But would that have benefited the patient? No, it wouldn't have. So we need to maintain that sort of structure in some form, including placebo. And then part of that discussion is to say to patients that if you're involved in a clinical trial, you'll do better than the current standard of care on average. And also to put pressure on industry to say that there should be an open label extension study after the completion of the trial so that everyone gets access to the active medication.
It's really important. I can't speak for here, but in US, maybe it's me, I'll really say really negative things about companies if they're not willing to do open label. I think that doesn't meet today's ethics for patients who are willing to join a trial. And so to the point where I might not even want to enroll patients in a trial that doesn't include that phase.
I'm not working directly in clinical trials field, so please correct me if I'm wrong, but I have two questions. So one is following up with what Matthew mentioned before. When I look up the clinical trials result, we have like different ways to measuring the data, the results. So like even with the controls group, we have different ways. We have placebo people don't have the treatment. We're using riluzole, we combine it with riluzole and creatine, and then different drugs, or even with the same drugs, we have different results. And then when I look at it, it's really hard to compare different trials and then giving us like idea what is going on with the patients. Or for example, with the neurofilaments, like not many clinical trials actually having the data that using neurofilaments, light trend. Most of them having like heavy trend that I haven't heard about before as well. So my question is there any way that we can standardize our protocol to design a clinical trial to make it more, I don't know, standardized across all the clinical trials that everyone follow, what kind of test and measurements, so like we can compare it over time? And my second question is, when I look up online as someone who don't work in clinical trials, the only public database that I can access is the clinical trials government. I'm not quite sure is that the right place to look at, but when I look like over 100 drugs being under clinical trials for ALS, only like about 50% of them have already published the data. So we can look up the data and assess it and then compare with what we're doing in the lab. But then we don't have like the 50% left. And then we don't know what is going on in clinical trials and how we can go back in our preclinical stage.
Can a person answer that question? Can one person, because we are running a little bit out of time.
So I think in terms of standardizing clinical trials, I think that is coming. There tends to be fairly standard inclusion-exclusion criteria and primary and secondary outcome measures. I think it is important, though, in new trials to embed experimental medicine so that whatever the outcome of the trial, we learn something about the disease and that therapy. So trials may vary in what embedded experimental medicine there is, but I think standardization is gradually happening across different countries.
Start having to wrap it up, but I want to end on one question for the panel, which is if there could be one thing that we, you can define we as any way you want, patients, hospitals, governments, the scientific community, the media, could do to change and improve things, what's the one thing that would have the most impact that we could do ourselves in some way, or a government do this, anything like that? We might start with Susan, who is looking sheepishly.
Yeah, I might have answered this differently some years ago, but I think the thing that has come to the fore in recent years for me is how important advocacy is. And Kate touched on that earlier. And I think all of the things that we have achieved to date really started by either individuals or small groups of individuals, be they patients, families, philanthropists, clinicians, researchers, you know, being passionate about some aspect of improving life for people with MND. And people themselves, the lived experience group, I think now have taken that banner to themselves, and that's a great thing to see. And I think they, you know, they're very good at telling us what's wrong in pretty plain language, which is needed. But they're also, I think, I'm increasingly aware of how much they're actually supporting us and encouraging us to do things better, because they kind of see that we all need to succeed. And I think patient voice and us all being advocates is probably what's got us this far and will take us forward.
Sorry, it's multi-pronged. It's health, it's always multi-pronged. There's never one approach. If you get one thing, you concentrate all your efforts on one thing, you won't be doing the patients the favour. You actually need to be looking at it more broadly.
Yeah, I'm not a good advocate for reductionism, I have to admit, but if I have
what I do for a living.
Yeah, well, that's why you get the smart suit. But the one thing I would think we could all advocate for is data. And so I would like every one of us to be contributing data about what we do to places that can be merged.
As an immediate thing, making aged care fair and funding all the supports that people need while they're waiting for better treatments and a cure.
One thing, I'm not sure I can come up with one thing. I totally agree about advocacy. And I think patients are superb advocates for doing something about this cruel disease. In the UK, you know, famous sports people, Doddy Weir, who was a Scottish international rugby player, Rob Burrow, Stephen Darby, have all gone to 10 Downing Street to the Prime Minister and the Secretary of State for Health and made the case. And they actually got Boris Johnson to promise £50 million over 5 years to develop this national institute. So I think patients having the courage to make the case for the condition that they're facing and their families are facing. I think also increasing access to trials for patients. So it disturbs me to hear about New Zealand. Why can't New Zealand be included? So somehow finding ways of enabling countries that maybe haven't been traditionally the go-to place for big pharma companies to be included in trials. And networks can really help with that. So NCALS has set up a lot of European centres who perhaps weren't traditionally taking part in trials, they now are. So, you know, setting up networks can really help with that and bring new centres on. So that's a long-winded answer. I don't think there's one thing that I could.
So just echo the advocacy, patient strength, but I think The one thing that I think would be the game changer would be going back to yesterday's presentation from Amar to identify the match that goes in and triggers the whole process. That would be the game changer. And in fact, this debate goes back to Charcot in the 1860s. It was a debate of function versus structure. And a lot of the studies that we're seeing presented are very TDP-43 orientated, but is that the downstream process? So should we be looking further upstream? And I suspect it's a molecular functional change, and that would totally transform the outcomes for patients.
Yeah, so I'm sort of in the same vein. It's maybe not one or two, but it comes down to what I think is money, people, and collaboration. So those are three. That's the first domino. You have them all working together. That first domino, the start point, everything follows in a very abstract way, ultimately to the patient. So to your pressure on us is that's how you get to that word cure. Starts with that. In the United States, now, and that's the core. And in the United States, we have the added advantage. And I don't know all the countries well enough, but philanthropy plays an enormous role. In some countries, it's sports. Certainly, as the United States, but in the United States, philanthropy is so strong. It starts with the people who really make a lot of money, the CEOs, the billionaires, and they have made a push in ALS in the United States, and that as such, that's the spark that also pushes that domino. So,
Well, thank you all. I think despite the first question and the reality of it, there's a lot of room for hope and there's a lot of direction to make improvements and find that breakthrough. So thank you all for your time.